Stanford University.
It's my great pleasure to welcome my friend Dr. Thomas Eschenhagen here. Thomas is currently Professor of Pharmacology and Director of the Department of Experimental Pharmacology and Toxicology at the University of Medical Center Hamburg. He's also the chairman of the board of directors for the German Center of Cardiovascular Research, or the DZHK. I think for those of you who don't know, the DZHK is probably one of the largest consortium in Germany, contains 7 major sites and additionally multi-site. And basically the purpose is to accelerate translational research in Germany. And I think some of the junior faculties abroad have been able to get positions through the DZHK as well. So Thomas is, I would say, best known for his work in terms of the tissue engineered heart muscles. Essentially, he's the pioneer, the discovery back in 1994, first using primary cardiac myocytes. And right now, I think as many of you know, have switched to using human ESL and also human iPSL derived cardiomyocytes. He's also been a leader in the areas of beta-adrenergic signaling and the pathways involved in heart failure. I think the work that he's doing, in my opinion, is very relevant because when you make these tissue patches, not only can it be used for regenerative medicine, for example, to create a patch around the heart, prevent the heart from dilating, but also using these patches for high throughput or medium throughput drug screening, for long QT testing, to understand the disease mechanism, understand systolic, diastolic functions. And in recognition of his discoveries and his contributions to science, he was awarded the Frankel Award of the German Society of Cardiology back in 1997. And then recently received the Outstanding Investigator Award of the International Society of Heart Research in 2012. And he's also currently the president-elect of the ISHR International Society. So thank you very much, Thomas, for being here.
Thank you very much, Joe. First, for the invitation, for your hospitality, and showing me around here. I'm really impressed by Stanford. It's actually the first time I'm on campus. I mean, everybody knows, of course, Stanford, but it's really a stunning place. I had very pleasant discussions yesterday already, but also today. So it's really a pleasure to be here. Given the time, I probably rush through some pictures pretty quickly. Do you have a pointer or something? Oh, we have to go like that. I start with a little confounder. It's in the drawer. Yes. Okay. Okay, so let's just be opened. If there's battery on. Yes. Okay, great. So a little confounder. We sell this technology nowadays. Okay, so I would like to start with a little challenges, because I think it's time to talk about some of the problems as well, and also the motivation behind this type of research. Then introduce you to this technology, engineered heart tissue, and then come to some cardiac repair work. So when I started my research, which is more than 25 years ago, I started in heart failure. And this is a non-complete list of all the drugs which failed actually in heart failure, starting with the positive inotropes and many other drugs, and we don't want to go into much detail. But many of these drugs really looked exciting in preclinical work, also the anti-inflammatory drugs, but finally they either did nothing or they even increased mortality. So somewhat, despite the overall success in cardiovascular research, heart failure isn't such a success story. And we're all happy that we now have, after 20 years, the first new drug, the LZZ. Okay, so there's something wrong or something difficult. And one of the things, I think we still don't really understand what's going on in heart failure. Despite beautiful work, which originally started with Mike Bristow in 1982. I'm sorry, I don't know how to get away with this bar. Don't understand my computer anymore. So it's just a bar, it should be not there. Anyhow, Mike Bristow is really a pioneer here. I tried yesterday evening. No, don't ask Cortana, then it's going to really into trouble. Yeah, chart slide.
Never mind. Sorry.
I don't know.
Technical difficulties.
It doesn't matter, so don't worry, sometimes they're interesting. Importantly, heart failure phenotype, because if we are talking about an in vitro phenotype, what exactly is it? It has been studied mainly in these trabeculae from explanted human hearts. And everybody would expect heart failure to develop less force than a normal heart. But if you go back to the old literature, it's not so easy. This is one of the first data by Feldman and Judy Gwasmi, 1987. A little bit unphysiological conditions, as you see here. But if you see the calcium response and the maximal force, absolutely no difference between terminally failing and non-failing hearts. And that has been reproduced by many others. So the maximal force production is actually unchanged in the failing heart. What is consistently different is a reduced beta-adrenergic positive inotropic response. So this is heart failure, this is non-failure. You see a marked reduction. That has been reproduced by many, many labs. And the third one is a delayed relaxation. If you do this at 33 degrees and low pacing frequency, you see a delayed relaxation. But here, Hasenfuss, Alpert and Mulieri's study, with increasing frequency, time of relaxation, time to peak, half relaxation, absolutely no difference between failing, again, always terminally failing versus non-failing. So even this parameter is not so clear. But what they found in this study, and that actually I think was somewhat paradigmatic, they showed that if you increase frequency, there is this positive force-frequency relation in the non-failing human heart trabecle, but no, or rather negative response in dilated. That looks very dramatic. It has never been shown again so beautifully. And if you go back even worse, even more, this is now 1966, Edward Sonnenblick, they did a very interesting study in intact right ventricle of men. So they implanted force transducers on the surface of the heart and stimulated at low rate, 79, and then went up to 125. And what you see is here, upper is force goes down, and then interesting, over 5 beats, normalizes. So what is changed, and it's not increasing. So what is changing is, you see the kinetics is changing, and thereby the df, the deviation, that's increasing, the velocity. So this one looks rather like a velocity rate response. And this is the statistics here. You see no change in force, but an increase in velocity. Because, of course, to reach the same force in a shorter time, you need more velocity, higher velocity. So this, I think, is a major change. And the group of Hasenfuss later did a similar thing in a patient now with dilated cardiomyopathy versus control. And here they measured dPdt with a catheter. And you see the difference in maximal dPdt, and with increasing heart rate, no increase in dilated, and a slight increase in control. But again, this is not force. This is velocity. And the increase is minor. So I'm still not so convinced that this decreased force-frequency relation is really one of the hallmarks. To put this together, there are several changes described in isolated muscle strips, or sometimes isolated myocyte, and there are, as you know, a number of molecular alterations explaining some of this. But again, maximum force unchanged, Frank-Starling mechanism unchanged, force frequency, well, probably reduced or nothing. A lot of question marks here. It's just not consistent, even in the human heart. So my conclusion is there's a large variability in defining the human heart failure phenotype in vitro, and it has to be taken into account that all we see in these terminally failing heart is, of course, something in the end of the development. It's probably not causal. It's more like a stereotypic adaptation to pathology. So what is causal? Well, we often don't know, and you know that in, for example, dilated cardiomyopathy, there are numerous reasons for the heart to go wrong. But one of the paradigms which seems to be, let's say, kind of consistent is that the normal relation between calcium and force at the level of the myofilaments is changed. If there's an increase in calcium sensitivity by mutation of the sarcomeric proteins, it's often a hypertrophic cardiomyopathy phenotype, and the opposite is like, that would be a very simple paradigm. Everybody knows it can't be so simple. At least it's something to work on. And that would predict that a typical dilated cardiomyopathy phenotype of a muscle twitch looks, compared to the normal in black here, like that. And the hypertrophic should be more force and probably also delayed relaxation, because calcium needs time to dissociate from the myofilament. And actually this paradigm is, I think, nicely promoted or supported by the data of Jennifer Davis and Jeff Morgentine and Joe Wu was also part of this study, that the main difference they found was not so much the calcium, but the tension force interval. So this is an interesting paradigm to test in the future, and I think it could be a nice basis for work also with iPS cells. Okay, so challenges, to make it simple here. Overall, I think one of the challenges is that heart function is only, I think, discretely changed. It's not a major, it's not like the heart doesn't work anymore. It's just a little deviation from normality, and that makes it difficult to study in vitro. We have to think about the long-term cause. It's a very, in general, slowly developing disease. There's a systemic dimension, and it's interesting to know that all the therapies are not primarily attacking the heart, but the vasculature, the neurohumoral regulation. It's not so much the heart muscle itself. And certainly, when studying all this, one of the problems is species differences. And I would like to make the point that phospholamban is a very good example of that. Many of you will know that this little regulatory protein, which is normally inhibiting SERCA, the SR calcium ATPase, so it's regulating diastolic calcium uptake. This little protein, if you knock it out in mice are perfectly happy. They've increased contractile force all their life long, but they have no decreased life expectancy, so they're pretty normal. In striking contrast, in human, both heterozygous and even more so homozygous phospholamban null mutations lead to dilated cardiomyopathy. A major difference apparently between mouse and man. And I think overall, phospholamban is a very good example in general for from where I want to come. And that's shown in this study. You may know that in the Netherlands, not this mutation here, which is a null mutation, but another mutation, the phospholamban R14del. So one codon is lacking, so one amino acid is lacking, and then there's a stop by this mutation. This is a very common founder mutation in the Netherlands. So that allowed people like Arthur Wilde and this group to study more than 400 people with this single mutation, all heterozygous. And the important point, I think, is shown here. If they check the standardized mortality ratio compared to norm, so one would be the normal population mortality, and this is the age group from 15 to 24 to very old people. You see that, and that's information in this paper, before 15, absolutely no risk whatsoever. A completely normal risk for death. But then, nothing here as well, still around 1, but then, this is men and women, and then in this age group, 25 to 34, here you have a 5 to 7-fold increase mortality rate, and then the older the patients get, probably you lose some here, the less it went down. So altogether, despite being clearly a disease gene, the standardized mortality ratio is only about 5-fold altogether. sorry, maximally increased 5-fold. The overall mortality only increased 1.7-fold. So this is a disease gene. This goes along with ventricular arrhythmias and LV dysfunction, but as many of these mutations, it has a very incomplete penetrance, which explains probably part of the problem. And it's numerous mouse models have been made with this mutation, or translating overexpression in a wild type, translating overexpression in a knockout background, and still not quite clear what happens here, whether this acts as a dominant negative protein or other reasons which are even less defined. So I think this is a good situation to look for another model, and obviously the IPS model developed by Yamanaka could be a way to go. And we all know that this lab here is very active in this area. And I like this article of Joe with the personalized medicine, because that could be a very nice answer. Because if you go in these families with such a different risk, it would be important to tell a family whether this specific mutation is a very risky one in this context of the rest we don't know, or less. So pros and cons. Of course, the pro of doing such things is iPS-derived cardiac myocytes would integrate the entirety of the genomics, the genetics. There are less compensatory mechanisms in vitro, so it could be more sensitive, this assay. And of course, you can do stress tests in these cells. So this is all a pro. On the other hand, you can ask, well, if these mutations in patients need 15 years to be relevant, do we really have a chance to see this early in vitro and in this in vitro phenotype? And is the effect size of this single mutation really higher than all the technical variability which we introduce in this model? And again, what is exactly the cardiomyopathy phenotype and what are we looking for? It has been done, and somehow Joe told me that Dr. Karakikis is now here. It's been done in the group of Roger Hajar, and what they found is this is a calcium transient in a wild-type cardiac myocyte. This is a calcium transient in this from a patient, and you see this arrhythmogenic phenotype, and this could be corrected genetically, the R14del, and in these two different cell lines, and then it was gone, so indicating that this arrhythmogenic phenotype is indeed due to this mutation. Unexpectedly, though, I would say, in this paper, I would have expected, and probably the authors themselves would have expected a decreased SR calcium content. What they show here is that in the diseased line, the caffeine response, meaning SR calcium content, is increased compared to the corrected, rather arguing for more circular function. It's difficult. And of course, if you have a look at the cells, this is an immunofluorescence from this paper, these cells are very immature. And here we come to a problem, and what to do it. I think one solution is very high numbers, good statistics. A second is high content, high precision. Third, both together, and of course, gene correction as has been done. And so we think that in terms of high content, these engineered heart tissues which we developed over the years, very early models here in chick embryonic cardiac myocytes, then here this ring model in red, could be one answer. And I just want to introduce a variation of that method, which we developed some years ago, which we think is really a good way, easy way to go. It starts with making rectangular casting molds in an agarose mold in a 24-well plate. Then we put in these silicon racks with 2 silicon posts each. And then we inject, or we pipette the fibrin and the cells in, and then we transfer the silicon rack with this fibrin block and the cells to a fresh 24-well plate. So that's very simple, looks like that. And then what develops is this muscle strip between 2 flexible silicon posts. And what you see, maybe, I hope, is that the post is a bit deflected. And that means this tissue, while developing, is always under mechanical strain from these bent silicon post. And that's important because it leads, this is histology of this developing tissue, it leads to a strictly longitudinal orientation of the cells. You see as well the marked remodeling of the gel. This gets thinner all the time, meaning the tissue gets denser. And if you wait long enough, in the rat, it really looks like a hard tissue. We never get it so nice with a human yet, but with a rat, it's really quite nice. And this is done in a 24-well plate, and you can just see here these little muscle strips in the bottom. And then we just constructed a machine where we put these 24-well plates in the incubator under controlled conditions, and then we had a software developed which directs this camera to move on top of this incubator with a glass roof and determine contractility. And that's how it looks. So the camera sees the spontaneously contracting EHT and follows the ends over time, and then you see this contraction pattern over time, and then you can calculate easily, because we know the elastic properties of the silicon. We can develop force, contraction velocity, relaxation velocity, times, and rhythm. Okay, and then of course you can do this with IPS. You can do it with all types of cell. We follow the same protocol. You see here, it's initially we followed the protocol of Keller, and then we got some inspiration from Burridge and Wu and others. And I think it's a pretty standard protocol. We don't use a Kier compound. We use a activine BMP4 FGF protocol. And if everything is fine, it looks like that. I have to admit, though, it is not always looking fine. And that's sometimes putting the entire lab into depression. And I'm sure we are the only ones. If it works well, and in general it works well, we can make hundreds of tissues per week. And then it looks like that. So these are all the cells. And so I just show you some impressions of the method. Here, for example, Ingram Mannheit just monitored them overnight. And you see here force, frequency, time to peak, time of relaxation. It's a very stable system. There's hardly any variability. This is n equals 4. These are standard deviations. So this is very robust. So the EHT time part of the coin is not the problem. The cells are sometimes the problem, but not this part. What we learned recently, in former times we measured calcium by fluor or fura. It's much easier to use a GCAM sensor. You see, it's a really strong signal. You see as well, some cells are blinking without being connected, but in general, it's a synchronous, nice beating. And you see here the signal-to-noise ratio is really dramatic, much better with fluor. This is a calcium signal, this is a force signal, and we have a setup to measure it simultaneously. Okay. Of course, what we expected is that this form of culture makes them more mature, and that you know that this is a major problem in the field. So we did some experiments. This is, for example, actinin staining. You see it's a dense tissue, but you see as well the red dots here. This is connexin 43. That's not normal. We would like to have them in intercalated disc. We never really clearly see it's always more distributed all over the place. So partially nice, partially not normal. The same here. This is the macroscopic view. You see nicely the muscle structure. If you cross-section, you see nice dystrophin stained muscle bundles. You see MLC2V. They're almost all MLC2V positive, so they're really ventricular. Nice cross-striation, actinin. We talked about cell isolations from this tissue. That's less than perfect. And you see that the cells look not super happy, and they're clearly not really rod-shaped, but they're going in this direction at least. And we do see some little tubes here which look like beginning T-tubules, but it's clearly not a fully developed T-tubular system. It's maybe a start off, but it's not a normal T-tubular system. What we did then is a proteomics analysis, and we compared 2D and 3D. And everything which is on the left is more in 2D. Everything which is on the right in this volcano plot is more in 3D. And what you clearly see the major difference was an increase in mitochondrial proteins. And we verified them on Western blot, some of them. And the pattern, the rest was not much different. The pattern looked exactly like the development of the human heart as shown by Thomas Brown's group. This is mitochondrial increase in adult versus fetal, and the rest is a bit similar. So that looks like metabolic maturation. So we went on and asked whether these 3D engineered heart tissues also eat more fatty acids and do more oxidative phosphorylation. And the answer is yes. This is oxidation of C14 glucose, C14 lactate, C14 oleic acid on the right. Always compared negative control, 2D, 3D. And you see always the 3D produces much, consumes and thereby produces more CO2. Well, you can say that's easy because, of course, if they beat against elastic strain, they should perform more work and therefore they need more. But overall, in the very same experiment, the total glucose consumption was higher in 2D. So 2D consumes more because it's not oxidative. It's, I think, a very clear sign of metabolic maturation. And one of the questions is always electrophysiology. And here we had the chance to directly compare by sharp microelectrodes the EHTs with the human left ventricle. We get human left ventricle, that's actually quite nice now with our surgeons. I don't know whether it's the same here in Stanford, but our surgeons implant LVADs like crazy. A lot. I don't know where all these patients come from. LVAD is very popular, so we get always this, the biopsy, let's say, where they make a hole. So that's very nice. So now we have constant flow of left ventricular tissue in the lab. And you see here, the action potentials look actually very, very similar. This is clone 1, clone 2, and upstroke velocity is 250 volts per second. That's very high. It's major in this respect. You see as well the resting membrane potential is very good. So in this respect, they're pretty good. Surprisingly, I have to say, good. Okay, is it good for anything? We tested numerous drugs. I don't want to bore you. Just make one point. Many people measure contractile responses in 2D, of course, with ion optics or something. And this is a group from industry, Poynton et al. And they, for example, measured calcium in blue and phos in red in different concentrations of verapamil. Of course, they see a negative effect on both calcium and phos. And this is a nice concentration response curve, whether it's stimulated or spontaneous beating. But in their discussion, they say the following. Despite being able to detect changes in activity with negative and positive inotropes, the direction of response was not always correct. All positive inotropes tested except iso and epinephrine produced a response in the same direction as negative inotropes, which is, of course, not really what you want. Showing the problem. So we tested several drugs, and I just show you some examples. It's an average peak of 4 experiments. Red is intervention, black is control. So this is, for example, medium concentration of verapamil. This is an increase in calcium. You see a harmonious increase in force, no change in times. In marked contrast, a calcium channel agonist or a calcium sensitizer of the myofilaments, EMD, both of them also increase the peak force, but they have a marked negative lusitropic effect, so they prolong relaxation. And the opposite is true with cyclic AMP-dependent drugs like ISO or the phosphodiesterase inhibitor Rolipam. They shorten contraction. So this is faithfully doing what's supposed to do. And of course, one of the big questions in the field is, could we use these systems also for the detection of pro-arrhythmic drugs? And the Joe group provided nice data, and we tested the same question. And what you see here is a correlation between published IC50 values, so half inhibitory concentration on the HERC channel, one of the major potassium channels, and log values for the relaxation slowing effect on the EHT. The idea was here, anything which prolongs relaxation, anything which prolongs action potentials should also prolong relaxation. That's actually what's true. You see a quite nice correlation. So we are seeing relaxation slowing as a surrogate of APD prolongation, and that holds true quite well. But of course, finally, the real answer is how good is it all compared to other assays, and I don't know whether you're participating in that as well, Joe? Yeah? In this hazy, so we just got these compounds with some complications, but finally we got them, and we are doing so let's see. Okay, just to come back to the phospholamban. We tried as well to do this experiment, and we didn't come very far yet, I have to say, but the first experiments look pretty clear. So this is a contraction pattern of a control cell line, similar to what you just saw before. This is of a R14del carrier. You see much less force production and arrhythmias, and that's actually quite consistent. Fine, so that looks like the paper of Roger Jar's group. You see here this in the statistic, clear decrease in force, no decrease or no increase, no change actually of the kinetics. That's the peak. And then we said, okay, if there's a defect in phospholamban, you would expect a decrease or some change of effects to isoprenaline or response to isoprenaline. That's not the case. Above are in green the values in the control cell line. Of course, you see a positive inotropic effect of isoprenaline and a slight negative inotropic effect of addition of carbachol. You see here the times, and that looks more or less the same in the disease cell line. Actually, the iso response is relatively higher. I don't know whether maybe that's similar to this increased calcium content of the SR. And this is we do an overnight stress test by increasing calcium, and these are individual muscles for several hours monitored, and this is a RR scatter. So that's a I started 45. Okay, so quick. Arrhythmias. I wanted to show you some other things. So, okay, so this is just a big project. I thought it's interesting. Here we try to. It's an ERC-funded project where we try to make a clinical study with iPS cells, where we compare 40 probants and 40 patients dilated in HCM. We do all the clinical examination. We do skin biopsy, do 80 cell lines, and then we do everything blinded, and we hope that in 5 years we will find an answer whether we can find normal values in the controls and any specific deviation in these diseased cell lines. That's quite an ambitious project, and we think that maybe one interesting intervention would be this afterload stress test, that we not only see them under basic conditions, but that we stress these tissues and then hopefully provoke its phenotype. And that looks like that. Since we have these silicon posts, we can acutely increase the afterload by putting a metal brace in. And so this is a normal behavior. It's bending the post. And here it still beats, but it cannot really beat them because it's such a high afterload. So this we call in vitro tuck. And we hope that by this we can provoke a phenotype. So shortly, because I know some people are interested in cardiac repair, obvious question is, can we do cardiac repair with these muscles? And we think a patient with such a heart really has no option with any drug anymore. So we did a study with our EHTs. Here time we did a bit bigger EHTs, 10 million cardiac myocytes, 4 million endothelial cells, so 2 strips of these. And we did, and I think it's important, we did something like a clinical study here again with guinea pigs. We started with 250 female guinea pigs. We had a high mortality by cryo injury, a big cryo injury. Then 7 days later, we transplanted either EHTs, cell-free fibrin, or endothelial cell constructs. And then we tested 4 weeks later what happened. again, always with the echo. And you see the numbers here. Out of these 270, only 550, only 72 have survived because we had this big mortality here, but we had large groups. It looks like that. So this is a guinea pig heart from the front. You see here below maybe an infarction, and we try to suture these EHTs here above and below. 4 weeks later, they are all integrated. With micro CT, it looks like that. It's quite beautiful. This micro CT technology, we'll see here. Here you see the myocardial infarction, and opala, and there the EHTs across. Okay, so this is how it looks 4 weeks later, all under immunosuppression, of course. These are just 7 examples, and you see here, for example, the right ventricle, the septum, the left ventricle, and you see here below the large infarction. And on the infarction scar are these structures here. And they are of different size, but they are reproducibly there. Here this is very small, but this is very big. You can say, is it really left over? Or is it really graft, or is it left over? Of course, it's a good question. So we stained by a Q80 antibody, which is human-specific, and you see these red, this is this part here. These are red islands, and the red is not red. And you see nicely with this antibody that much of the cells in between are host-derived. They're not red, but all the cardiac myocytes are red. So that we are sure that these are human muscle grafts. And I would like to point out the following. If you do some calculations here, the guinea pig has a 2.5 gram, the LV 1.6 gram, we had a 22% scar size, it's 350 milligram, and 12% in mean graft size is 43 milligram. And if you calculate this to numbers, you end up with 1.8 million hips. It's rough numbers. But the input was 10 million, so it's an efficiency of 18%. And that's actually very high. If you compare this with data, for example, here of Chuck Murray, which I think is the reference at the moment, they saw similarly big grafts, but with 100 million per heart. And then, of course, you get 10 times more washout. So I think that becomes relevant. And just to make another point here, we were really surprised to see these big grafts. Look at this. This is the input. This is a cross-section of an EHT. It's about 800 micron, and you see it's not very full of cells in the middle. There's hardly any. It's just around these big grafts have a problem in the middle. But then, 4 weeks later, they are like that. And if you project this piece there, something must have happened. And what happened is a marked degree of proliferation. These myocytes proliferate really a lot. I don't know, yeah, you can see this nicely here, the brown nuclei. 15 or 16% Ki67 positivity, and 1 or 2% PH3 positivity. This is all a graft, actin positive, these are cardiac myocytes. This is surprising because the cells which we use to make EHTs, they proliferate at around 11%. So 90% cardiac myocytes, 11% of them are proliferating. But that's at the moment of EHT making. So this is day 0. So you have 30% Ki67, but then over the time of EHTs, the Ki67 positivity goes almost down to 0. So what happens is it goes down, and then in vivo goes up again. You see here, MSC2V goes up, MSC2A goes down. All signs of maturation in vitro, but then apparently the opposite happens in vivo. So I'm sorry, we're too late. They are vascularized, and they do couple. This is an example of a coupled EHT to photon microscopy. This is an EHT action potential and a graft action potential in green, host in black. At 4 Hertz, nicely coupled with a remote. At 7 Hertz, there's loss of capture. And this is arrhythmias in the cell-free and the EHT. And actually, we did not see any arrhythmias, neither with or without these EHTs. And this is probably the most important one. We had a good functional recovery. So this is fractional area shortening before injury. 42% goes down to about 26. And then in the EHT group, it almost goes back to normal. And there was no significant effect in cell-free and less, also not in the EET. Normally people show it like that. Looks much more impressive. The real data are like that. There is a normal scatter. With the SEM, you see here this quite dramatic effect in this group. So with this, I would like to summarize. I think it's easy to make sufficient numbers of human myocytes today and tissues. They do structurally integrate and vascularize. Electrical coupling, yes, with a little question mark. No evidence for arrhythmias and clear functional improvement. Why? It's really still a question mark. Why is such?
Stanford University.